Hello, everybody. Good night. Good morning for some of us, some of you. We are starting the webinar, The Emerging Science of Water and Its Applications. I'm talking here from Bali. We are having the World Water Forum and the alternative for going on the city of Bali here in Indonesia. And as we did in Brasilia World Water Forum, but also in Dakar World Water Forum in 2018 and 2021, we are here to advance on this spread of this very important field of research regarding water and molecular level of water. So this night we will have very prominent speakers, lecturers, to present a little bit about those emerging science of water. We are going to have a keynote speaker, that is Professor Gerard Pollack, and then some other lecturers from other parts of the world. We have a big time zone difference. We have people in the west coast of US, but also in Japan. I'm here in Indonesia, people in Brazil, lots of colleagues from Europe as well. So we are in a global network of people looking for water and caring for water and trying to understand water in a deeper level. And this is very welcome for the planet. So I'd like to introduce myself. My name is Sérgio Ribeiro. I'm the Director General from CIRAT, that is the International Center on Water and Transdisciplinarity. We are an institution that we are in the process of qualification as a Category 2 center under the auspices of UNESCO on this theme of water and within the International Hydrological Program. And I would like to start this webinar by inviting Professor Gerard Pollack, that is professor from University of Washington, and also the editor from the Water Journal. He's a great reference, one of the greatest references on this field of research. So thank you so much, Professor Pollack, to be today with us. It's very early on your place. Thank you to be here. And you have 15 minutes for your considerations. Thank you so much.
Well, thank you, Sergio. Let me share screen. One moment. I need the full screen. Sorry. Slideshow. Okay, thank you. Well, I'd like to talk about the fourth phase of water. And also, I would like to include a surprising role of light. I start with a sort of general question about water. We think we really understand water because water has been studied for so many years. I'd like to demonstrate to you that we really don't understand water. And I'm going to show a series of slides to make my point. First slide, well, clouds are made of water, little droplets of water. And if you were to climb on a ladder to the level of the cloud and take a pitcher of water and turn it over, it would descend right away. But the cloud doesn't descend. And so the question is, why? And if you don't know the answer, then you don't know all there is to know about water. Another one is Water Bridge by Elmar Fuchs. And you take two electrodes and you put a high voltage between the two waters, and a bridge forms. And if you move one beaker away from the other with a motor, the bridge sustains itself. And it sustains itself for up to 3 or 4 centimeters and indefinitely. Can you explain it? I think not. Here's another one. It's water dropping on water. And as you know, when you drop water droplets onto water, the two will coalesce instantly. But as you see, it doesn't always happen. And we've seen instances where the droplets can be suspended for a period of even 30 seconds or so before the two actually coalesce. Explain it? I think you don't know the answer. And here's a famous lizard from Central America. It sits on a limb, and it enjoys itself. And when it gets stimulated, it jumps into the water. And as it jumps in the water, it actually walks on the water. And because it walks on water, it's called the Jesus Christ lizard. How does it walk on the water? What sustains it? And if you don't know the answer, you don't know all there is to know about water. This one is my favorite. This is a trough or container filled with water, and some red dye is put into it for effect. And when you turn on the magnet, what happens is that the water moves from the center to one edge and the other edge. And this is a real demonstration that I saw in Tokyo. And it creates a region here so shallow that Moses could walk across the Red Sea. This sometimes causes or can cause difficulties, like this particular difficulties. The difficulty, if it splits, those who do fishing are not so happy about it. Anyway, I got my inspiration from this guy, Gilbert Ling, who passed about 5 years ago. And he had a point of view about water in biology, water in the cell. He said that the water was actually structured. In other words, the water molecules were organized in some way. I was really intrigued by this, and we got started. This was some 20 years ago or more. I was so intrigued by this because if he was right, a lot of biology would be wrong. And so we set up an experiment, and it was based on Gilbert Ling's idea. And his idea was that inside the cell are solids, like protein, for example, and here are water molecules. And his idea was that the charges sitting on the surface of the macromolecules could organize the water molecules. And chemists will agree that maybe one or two molecular layers of water might be organized, but Gilbert was arguing that no, it's not one or two layers. It may be 50 or 100 or even a few hundred layers. And I will show you that basically he was underestimating because the number actually can be as large as hundreds of thousands of layers. Anyway, the idea was that this acts sort of like a crystal because all the molecules are organized. And so we were looking for a kind of, when we started our experiments, a kind of crystalline uh, region of water. And because it's a crystal, it excludes solutes and particles as crystals do. So we were looking for some region of water where solutes or particles were excluded. And we found it. So this is a chamber. And in the chamber, we put a gel. And we also put water plus particles. And these particles are actually microspheres, little spheres that are used in a standard way in all scientific endeavors. And we noticed that right next to the gel, there was indeed a region where these were excluded. And it was more profound than just that, because if you watch the video, this is over about 10 or 15 minutes, you can see that there's something, this water region is growing somehow and pushing out these particles. And because the particles were excluded, we called it the exclusion zone, or easy water. Easy is easy for people to remember, and so easy. And later, when we found that the water here has properties and features that are so different from ordinary liquid water, we began calling this water fourth phase water. In general, we found through many experiments that many hydrophilic surfaces, that is water-loving surfaces, generate these exclusion zones, and many solutes and particles are excluded. So question is is this water, this easy water, is it really different from bulk water? And this is a summary of the evidence, and the summary was actually put together as a slide about 10 years ago. And by now, the number is probably double this. I'm not going to go through it because it's more information than perhaps you'd care to digest. But basically, for example, EZ has negative charge, and we measure that using measurements of electrical potential. So these are the various measurements, and these are the observations. And the summary is that EZ water, fourth phase water, is completely different from ordinary liquid water. So one of the important features that we discovered is that it has a battery-like feature. So think of this as the gel or polymer, hydrophilic material, and this is the water. Starts with water, and it winds up being an EZ and ordinary liquid water beyond. We stuck electrodes in here, and we found, to our great surprise, that this was negatively charged. And this region we found is positively charged. When you have negative next to positive, it creates a battery. So this is water, and it's water that actually has energy, potential energy of a battery. And the way it works, we found, is this. We start with water molecules. See, initially, all of these are just ordinary water molecules. And the water molecule looks like this, two negative charges on the oxygen, and the hydrogens are here. And if you supply energy, and I'll mention that in a moment, the kind of energy, it breaks the bond. And these positives wind up here. And this is net negative, and it winds up here, building this exclusion zone or fourth phase water. So question is, if this is a battery, what is it that charges the battery? Because batteries always run down and you need something to charge it. And we found that it's actually light. Incident radiant energy is what charges it. And it was actually an undergraduate student who found it, and he was experimenting with a hydrophilic material, Naphion, next to water. And this is the usual experiment where the exclusion zone is here and the microspheres that are excluded are here. And you can see that the boundary is always parallel to this nucleating surface boundary. However, he took a lamp that was sitting on the bench and illuminated this region here, and he noticed that the exclusion zone expands where he has it. And we found in serious experiments that are published that the most effective is infrared energy. And infrared is just all over the place. So the energy is amply available to build this. And so, which means that if you have hydrophilic material next to water, you'll always have some easy water. And if you add more infrared energy, then you get more easy water. So it's a very simple paradigm. So energy for easy buildup or fourth phase buildup is powered by photonic energy, which charges the water battery and orders the water. Now, this applies in the cell too, because as I demonstrated in my book called Cells, Gels, and the Engines of Life, what happens when cells are activated is they go from a quiescent state where the water is ordered to an action state when the water is liquid water and back again. And so this quiescent state is basically filled with easy water, filled with negatively charged easy water, and therefore it has energy. It has potential energy. Your cells have potential energy coming from the water. And so if you think about it, getting practical applications that come from this is getting energy from water and sunlight, which contains a lot of infrared energy. And so all you need to do is, you've seen this before, is put two electrodes in, one here in the negative, one in the positive. And in theory, you ought to get current enough to light or to run some device or light an LED, and which is what we did. Here's These are several chambers hooked up in series or parallel, in which we have Nafion and water and EZ and two electrodes. This is a switch, and here's an LED lamp underneath a magnifier. And if you turn the switch, voila, it lights. So we have proof of principle that out of water and light, you can get electrical energy. So I would conclude by saying that one of the major findings of the research is that we think of water as having three phases: ice, a solid, a liquid, and a vapor. We found that there's a fourth phase, that's the easy phase. And the structure, it turns out, I haven't had time to present it, but the structure is not so different from ice. And if you go from water to ice, you actually pass through this phase. And if you melt the ice, you pass through this phase again. This is all demonstrated in this book, this is the latest book, and it turns out to be rather popular. So I think I'll end here. I've used my 15 minutes, and thank you very much for listening, and we're really excited about what's going on here. Thank you, Sergio.
Okay, Professor Pollack, thank you so much for your brilliant presentation, as always. I would like to go straight to our other lecturer this night. So hold on, please. Everini, can you listen to us? I'd like to introduce you. Just a minute.
It's not Pierre who was supposed to go before me?
So I cannot find your bio here. Yeah, but Everini van der Kratz, that is from World Water Community, and a leading scientist on this theme of water molecule and researches related to water. So welcome, and you have 8 minutes for your presentation. Thanks.
Okay. So, thank you, Sergio, for organizing this event again, because solving water challenges really requires global collaboration amongst all people and organizations, and that's what we're doing here. So this is great. And in these 8 minutes, I'd like to introduce everyone to the World Water Lab initiative, which is all about empowering water science concerned with the emergent properties of water through collaboration. Yeah, I'm Everine. You just introduced me, so I don't need to say that. What are these emergent properties of water, actually? So they're phenomena as yet unexplained by conventional science, such as what Jerry just mentioned, new phases of water, for example, the exclusion zone, water memory, structure, organization, coherence, unexplained behavior of water, for example, at interfaces or on nanoscale, properties of ultra-high dilutions, for example, homeopathy, effects of vortices and geometry on water, water programming, for example, storing, transmitting, and retrieving information, effects of sound, energy, electromagnetism, and frequencies on water, and the relationship of water to intention and consciousness. And many of these water treatments, for example, vitalizing and energizing, which are related to these emergent properties of water, have applications in health, agriculture, industry, and more. And I'll point out a few at the end of this presentation. So who are we? We are a community of individuals and organizations coming together to advance the as yet unconventional water science related to the emergent properties of water. We are scientists from different disciplines and backgrounds, because water touches all disciplines, as well as entrepreneurs, inventors, therapists, consumers, artists, designers, indigenous communicators, educators, and more. And these are all members of the World Water Community and support the World Water Lab initiative. Our common intent is to thrive, be respected and well funded, while bringing forth discoveries and solutions related to water as a result of deeply understanding and living by the essence of water. And our aim is to enable and facilitate cooperation in a safe and trusted environment with different levels of access, because currently the nature of our field still requires this. And as a metaphor, we use the tree, and this access to the tree ranges from closed to the public, which is in the soil, and open to the public, which is in the crown. And we thrive when this emerging water science is indispensable. The sap in our tree flows when we bring our solutions and commitment and expertise out in the world, and then we receive more manpower, project requests, and funds. In the soil, we do our work, such as fundamental science, applied science, and project development. And we have meetings to discuss all these things together, and we work on specific projects which should deliver high quality water science, and these projects can also be requested from people outside the World Water Lab. So in the Crown, we spread our work, and we have four main branches, which is awareness, education, knowledge hub, solutions, and implementing them. And we are organizing webinars, media, and awareness events, developing courses, university curricula and school curricula, building a World Water Library, sharing our publications, and implementing pilot projects and quality marking existing solutions. So in short, our aim is to bring the huge potential of the emergent properties of water out into the world and expand our field and become a strong voice together. Milestones achieved so far are that we sent out the first invitations to scientists in September 2021. We had the official launch in October 2022 at the Water Conference. And so far, 28 scientists have joined, excluding the supporters and others whom are part of the World Water community. And we have 18 general online group meetings so far, where we discussed our research, such as results, protocols, methods, theory, and models, and specific topics, and how to develop our field further. And so far, we have one active focus group dedicated to creating an educational textbook, which started in September 2023. So why are we doing this with the World Water Lab? It's to connect and discuss to further understand water, to use the enormous potential that we collectively have, to cross disciplines as water touches them all, to share equipment and expertise because we need a lot of different techniques to study water, to share contacts, to replicate findings and to build a solid science, to share protocols, to develop new scientific methods that fit the nature of water, because water is very dynamic and always in interaction with its environment, and to bridge between scientists, businesses, and consumers, to educate, as I said, to share the overhead and increase our efficiency and optimize the use of funds, and to collectively support and fund fundamental water science, to save, keep, and collect knowledge of pioneering water scientists, to build a living World Water Library, and to address the urgent need for young scientists to join this amazingly interesting field. And of course, to address the opportunity, because this field of water science has solutions for current challenges in climate, health, agriculture, energy, and industry. I'll come back to some later. So as I said before, the World Water Lab is embedded in the World Water community, and water community, that is you, because you are water. So we are all part of the World Water community in basis. And if we understand that we are water and that water is everywhere, we have the power to transform the world. The World Water community is dedicated to the global to local healing and restoration of our planetary and internal waters. And we invite you to come drink with us, because you will find inspiration, knowledge, in-depth conversations, solutions, and projects, events, as well as opportunities for inner reflection and personal growth. The World Water community offers a lot of tools and shared workspaces and services to bring this vision into reality, and of which the community platform is the main hub. And this is all to increase your impact, efficiency, and effectiveness. So you can come and explore this for yourself at the worldwatercommunity.com. And now related to the UN Social Development Goals, our members, most of them are deeply aware that understanding water brings solutions to many of the world's problems today. And cutting edge solutions are already being implemented, and they are based on new insights and knowledge we have about water. I will mention just a few here. For example, we have impact on improving water quality, bringing back rain even, reducing water use in agriculture, which is very important. And we can do that based on natural solutions, which the World Water Community Group is mainly doing, or new water science-based technical solutions, which come closer to what we are doing at the World Water Lab, such as the natural solutions of living machines of John Todd, restoring the water cycle in general, syntropic and regenerative agriculture, or new types of water filtration and revitalizing or informing water via energetic, electromagnetic, geometric, or dynamic water treatments, and much more, actually. So come drink with us because we are water and water is the solution. And we could really use more support and active members because it's a big endeavor that we've embarked on, and collaboration is key. So thank you for your attention.
Okay, Everine, thank you so much for your presentation explaining World Water Community and Water Lab. And you do a wonderful job with those two institutions, fostering the knowledge on water and connecting researchers and general public to take care of water. Very good to have you here with us. I'd like to continue here our webinar, inviting Professor Homiana Tsenkova. Professor Homiana Tsenkova is a professor at Kobe University. She's the head of Aquaphotomics Research Field, chair of the Aquaphotomics Society, and also chair of the International Aquaphotomics Society. So, Professor Homiana, you have 8 minutes for your presentation. Thanks.
Thank you, Sergio, for the introduction. Here we are. Okay. I was, first of all, congratulations on continuations of your initiative. I think it's a very good way to encourage more people to learn about water, to realize how important it is for our life. I will talk about aquaphotomics, which is next year we'll be celebrating 20 years since the announcement of aquaphotomics. I did it in 2005 at the International NIFAD Spectroscopy Conference. And we'll be having the 5th International Aquaphotomics Conference in Kobe in May 18. So you're very welcome to come. And I was trying to the emergence, I like very much the name, the emergence of water science. And it is really the emergence of interdisciplinary water science. Because this is probably the only science that is so much interdisciplinary. Aquaphotomics is, I see it as not only a multidisciplinary science, but also technology and educational platform, because it has its applications already, and also it should be it's not yet. We're not there, but it should educate people. So aquaphotomics is my image about aquaphotomics is like this. So we have so many omics disciplines, and we still could not solve the problem with human health, for example, and so many related to biological systems, for example, or water-based systems. Why? Because we cannot have real-time information for each of those in each of those disciplines. But actually, what is the platform? What is the field? The quantum field, actually, that connects all these elements, and being the ultimate sensor, what I've started seeing in our results in experiments, it reacts to any changes in the environment and reacts with any molecules or substances. So by dynamically observing the water changes, the water morphing, we are able to model and identify other substances and their dynamical changes, not only quantitative changes, but also structural changes. So the concept of aquaphotomics is that water is a collective mirror and at the same time amplifier, because it is water molecules are all connected. As everyone just said, we are all connected because we're water. So, but what water gives us is, for example, instead of analyzing that amino acid as one by one in protein, if we look at the water, we can even identify a small structural changes in protein, which is unmeasurable at the moment with other sources. How do we measure water? We use light. And it is a kind of continuation of Jerry's talk, the surprising light effects. So it seems that we can do lots of things with light, but this is why water connects everything, because, and why water is amplifier, because all water molecules are connected. So these are the instruments that we use. Recently, we have started using very small spectrophotometers that measure light absorbance at different frequencies. But at the same time, we can influence water with light at different frequencies. Jerry mentioned that infrared light can make easy zones, yes. Near-infrared light that we use is even higher energy. So that light could make and break also hydrogen bond. But it makes hydrogen bond because it's adding energy and makes water a battery, but at certain point, the water structure also, water restructures as well. And this is another small part of the instrument. The idea of water being, of having dynamic, monitoring dynamic changes of water gives immense potential, enormous information for understanding of the whole system that involves water. And this is the core of aquaphotomics, that number one, water is the only substance that absorbs any energy of electromagnetic spectrum. And water has different structures with different number of water molecules, but they could be identified by the frequency of a vibration, exactly by spectroscopy. So what we did, we identified exact frequencies respective to water molecular structures that are and structures are related to functionality. So by looking at this aquagram that we call the spectra of water, we could identify this liquid water, this is water vapor, for example. So we could identify also if this this is the band which is changing the absorbance of. It means that we have trapped water molecules, a single water molecule trapped ions, for example. These are very small examples. We have published papers already related to water absorbance bands we call WOMACs and water functionality. And this is our recent studies about we have identified each of those frequencies and its respective water structure and its respective function. So, and our recent we do monitoring of people listening to different sound frequencies or sound treatment, and these are the models. There's a beautiful correlation between time and dynamical changes of water. But when a person is perturbed by some treatment, like the guest number 3, you could see that it's highly influenced by sound. The guest number 3 doesn't react. So this is a way to follow people after meditation. These are all these 12 people's aquagram. So we could see everybody has a specific portrait kind of aquagram, but when they have a specific perturbation or treatment, the dynamic is very similar. So we talk about coherence, and we can identify all the structure with specific function on it. This is our recent study with after meditation. We could see before and after changes, similar changes for all our participants. So I don't have, I'm at the end of my talk, but one thing that really I wanted to share with you, these are the Weber telescope recent results that showed a water on another planet that has absolutely the same water absorbing bands that we have discovered. The spectral pattern is different, but what is there and the same structures that we know are there. Thank you very much for your attention.
Okay, Professor Romiana Tsenkova, thank you so much for your presentation on this so interesting and promising theme of research that is aquaphotomics. And now following our event, our webinar, I would like to invite Professor Pierre. Professor Pierre is the director from Edge Institute from Austria, and also professor in the University of Salzburg in Austria. So welcome, Pierre. Pierre, that it's important to make a remark made with other professors, a special issue on the themes related to water molecule, and he's going to present to us this night or this day. Pierre, it's with you.
So everybody can hear me. Good evening, good morning, good afternoon. Okay. So I'm not a professor, first of all. I'm still a postdoc, so just to make things straight. The topic that you can see on the screen, Hormesis and the Power of Water, came up quite after we have launched this special edition. The special edition that we started, actually we were invited to do that a couple of years ago, 3 years ago, has been closed by now, but we were able to gather quite a number of papers. And why hormesis on this particular topic that should actually deal with quantum biology. So currently we have 9 papers plus one. One is still pending. And we have the topics that cover distance signaling, hormesis, EMF exposure, low dose exposure, and quantum biology. And the common denominator is hormesis. And hormesis just in this graphic, it's just displayed here using magnetic flux densities, is that you have a high magnetic field that induces an adverse effect, and then at very low dosage it becomes reversed, and this biphasic response is classically described hormesis in a nutshell. So let's get started with the first sections of paper. This is paper number 10, which is still missing because we are waiting for the reviewers on their final judgment of this paper. It's about dry water matrix, dry water polymer matrix. It's a very controversial topic, but I think it's worth to get it out to the wider public and to get it discussed and shared with the others. The other thing is a new vision of the hydrogen bond. And in this case, we leave the first quantization, which is focused on particles, molecules, atoms, and look at it from a wave perspective. But this requires coherence. And in this particular contribution, the paper talks about phase angle correlations, which is the classical, let's say, low energy level state that has a better description, or allows a better description of the classical hydrogen bond that in the second quantization doesn't exist at all. It's just the tight oscillating capacity of the resonant attraction that keeps this stuff together and forms a coherent cluster, which then was commonly called in the past hydrogen bonding, which I think is no longer appropriate. So then we come on to the next sections of papers, which is glyphosate at low doses applied by the Brazilian group. And they could easily show that if you highly dilute this stuff, it's still toxic at those low dose levels, which means it induces abnormalities at this particular Now-play stage. You can see, hopefully you can see my cursor over here, but the antenna are missing. So, and the effects, interestingly, is not a gradual one if you dilute it, but you have a very strong response at a certain dilution range that fades off if you dilute it higher, and it fades off as well if you get it stronger. So somehow it's again a resonance effect that's a very intriguing effect that has been accepted and is published in this particular special issue. Another one is interferon studies published by the Slovenian group. And I think Igor will be talking about, maybe in a couple of minutes' time, about this particular topic anyway. So I don't lose too much time about it, since they have two papers in this particular contribution. One shares the communication signal transfer over distances, paper contribution number 1, and paper contribution number 2 is if you induce a mediator. In this case, if you use a secondary mediator, and shows that it still works. So you can have a couple of mediators and signal that the cascade of the information from one batch to the other without having been chemically in contact. It's just a physical contact, electromagnetic coupling in this case. The next topic is EMF applications. And in this case, we have a 50 Hertz magnetic field exposure and reactive oxygen species production in a zebrafish embryo. So the Daniel species is in the larval stage, has been exposed for 1 hour and 24 hours to various magnetic field intensities ranging from 100 to 800 microtesla, which is not really a hormetic approach, but still it is interesting if you use, for example, a 25 nanogram injection of this promoter in 1-year-old eggs and look at what it happens. Actually, it renders them more susceptible to the magnetic field. And this is then an interesting effect that you have a synergistic property in this particular aspect that was also considered worthwhile from the reviewers and has been published in this special issue. Another one is ultra-weak fractal EMF stimulation using cells in vitro. In this case, we used Haycat cells, keratinocytes, and exposed them to Malassezia, which is a fungi. And this fungi is usually not compatible to these cells, so they respond with a huge cytokine response, so the inflammation markers go all up. However, if you introduce just for 10 minutes a day, and we have done this for 4 days, 10 minutes a day, a fractal electromagnetic signal with a very low dose, in this case, we're talking about nanotesla range, so in this case, almost 10 nanotesla. The inflammation markers go down, and the resilient markers, in this case, HBD2, go up. So also this has been honored by the reviewers and has been published in this special edition. Then the final section is quantum biology, and we have one paper from the Rovianos group that we just heard about talking, and it's about superradiance. So how can cognitive processes in the brain be understood from a contemporary perspective? And this mechanism goes back, or it dates back to Carl Pribram, who back then postulated the brain as being a three-dimensional holographic network on various planes. And the various planes in this case have been abstracted as the neocortex, intermediate layer, scalp, what you name it. There are several, not just on these, because you can actually use also the pineal gland and other organoidal structures in the brain for that. So superradiance in this case comes from the Dykes theory and is nothing else as the wavelength relationship between two components, in this case particles, and their relation with the wavelengths of light. So if the wavelength of light is lower, or in this case smaller, then nothing really particular happens. But if the wavelength of this particular interatomic or intermolecular distance is larger, then you have two responses. One response is superradiance and one response is subradiance. Superradiance actually and subradiance are both coherent interference mechanisms that are used in this particular approach from Professor Pribram. And I take this particular contribution especially because if you consider two people talking to each other, as we are sitting here, right? I'm talking to you and you're listening to me, and I'm trying to convey a certain idea, crashing down my information pattern into a few words, which is the bottleneck. So I'm excluding information. It's actually talking exformation, and convey this to you as I'm talking right now. And you in your brain build up a new landscape. It's like a landscape that should match my brain map, so to speak, my 3D holographic brain map. And if these two more or less match up each other, then we can say these two brains become coherently coupled. And once these two brains become coherently coupled, then they can act as a unit. So then we have another paper here. It's the last one, the last contribution, which is the quantum electrodynamic ontomesis concept that was also honored by the editorial board and the reviewer board and accepted in this particular issue. And he talks about dropping the first quantization and focusing the second quantization as a coherent regime when the phase angle matches with the oscillating entities employed in this particular regime. So you have a couple of atoms or structures or molecules that oscillate in phase, then you cannot distinguish anymore the single atoms or the molecules, but you act as a cluster. And since this particular relationship is scale-free, you can have this observation not just on the microscopic scale, but also on the macroscopic scale. And you see this also in nature, and swarms of fish, swarms of birds, and even swarms of groups of people, individuals. So it's not just limited to biomolecules, organelle cells, and organisms, but also to social structures. And in practical essence, what is done is you can actually see how this subtle stimuli, if you have a very subtle stimulus, what you need is just a system under tension, and you apply a resonance frequency, and a Jardine effect just does that. You have a field grading, a very weak alternating electromagnetic field, and suddenly you have an oscillating or create an oscillating biochemical rhythm, just as the citric acid cycle. And with the citric acid cycle and the Jardine effect, you have a nice overlap. And this is probably one of the next issues that needs to be specialized in future papers. Another thing is that we have already heard about the Gilbert link and the association induction hypothesis, that a protein becomes only active in the conformational stage, producing something, when the water around it is in the decoherent phase. When it's in the coherent phase, the protein itself becomes blocked and is inactive. So it's a kind of on-off switching mode. It's like the next level of the epigenome. It's maybe an epiproteome, so to speak. So coherent coupling is not dependent on a signal intensity, so you don't need strong signals, but you need phase coupling, and this is not coupled to signal intensity, and this is probably the essence of this particular contribution. And therefore, second quantization, which focuses the field and not the particle, the matter-like driven perception of reality, needs to be envisioned, which is a paradigm change, of course. So the concluding remarks for me, and I hope I'm filling time, is you have the classical chemical hormesis with a lethal dose, toxic dose, and effective dose, 50%, marking denoting 50% in the pharmacological toxicological regime. However, the toxicologists hardly speak with the endocrinologists, But there's few, let's say, substances there in endocrinology that induce hormonal change and sex characteristics and features are expressed without being attacked by the organism, because this is at a very low subtle level, which doesn't actually induce an organismic defense response. However, if you go below that level, you become at the lowest effective level or the no effect level at all. There shouldn't be any response at all. However, when you approach zero and there is no substance involved anymore, suddenly you have the physical regime takes over. And this is a little bit, I know it's a bit odd to display it in this way, but the graph doesn't allow me to do it otherwise. So it's the chemical regime ends up here and the physical regime starts up down here. So it's between the zero and the zero, so to speak. But in this case, we can see, because of the hormesis response, not using chemical substance or just electromagnetic fields, that you can actually induce a drastic or an organismic-wide response. And this is, again, a focus or that underlines the relevance of second quantization in favor of first quantization. So, thank you very much for this brief slot, and thank you very much for the organizers to have me in. And I hope you enjoyed this little excursion, and maybe you have a look at the special issue that you can access. I will flip back to the very, very beginning of the page so you can access this particular journal. Thank you very much.
Thank you so much, Pierre, for this so interesting presentation. So many data and tendencies, very interesting, all of that. I would like to suggest that you share with us the link in order that we put in the YouTube for the people that part of our event can download and see additional information on that.
Yep.
Okay. Okay, perfect. And now going ahead with our event, I'd like to invite Professor Igor German. He's a professor of theoretical biology at the University of Ljubljana and the director of Bion Institute in Slovenia. So, Professor Igor, you have 8 minutes.
I don't have, you should allow me to share screen, please.
I think it's allowed. You can try it.
I see host disabled participant screen sharing.
Professor Igor, can you hear us?
Yes, now it is okay. It is the screen sharing is allowed. Okay. So do you see?
Yes, it's starting. Yeah, we can see it now. Go ahead.
Okay. So thank you very much for this invitation. I greet all of you. And I will just delve a little more into what was already presented by Pierre in his presentation, namely about the physicochemical research of molecular information transfer. And this means that we researched if the molecular signal that results from ultra-high homeopathic dilution can be transferred, and what time or duration or distance and activation means in this regard. So if I move on, okay. So in introduction, we know that more and more this research into homeopathy is becoming published. And the more we are convinced that we witness a real phenomenon, not understood or explained by established natural sciences, the more we need to have a solid scientific explanation for it. In principle, the so-called memory of water phenomenon, or something that we know from homeopathy or ultra-high dilution effect, can be based on matter, so-called nanoparticles, or can be something composed of specific energy, for instance, as presented by coherent domain theory or some other form of as yet unknown energy, and so on. Of course, it may be also a combination of the above too. From this point of view, it is very important to know whether the information of the ultra-diluted substance from the homeopathic solution can also be transferred to another liquid medium in a purely physical way, which means without a direct contact. This means without dilution. If it were true, it would speak in favor of the second possibility, of course, not neglecting the first. Such transfer of molecular information from one liquid to another without an intermediate liquid phase has already been a target of many scientific investigations. In this presentation, we will just show this, let's say, systematic research on this. And we developed a specific receiver solution to detect the signal, molecular signal. The molecular source of the physical ultra-high dilution signal used in the donor solutions was interferon gamma antigen, and of course homeopathically potentized. We paid special attention to how the transfer of the signal was affected by duration of exposure of the receiver to the donor, then how the distance between the donor and the receiver influenced the signal transfer, like seen here in this picture above, and how the transfer is affected by activation, which means shaking, versus exposure alone without shaking. So we have developed a specific methodology of distilled water, donors, control, receiver liquids, and so on. We used 4 different detection or measuring methods, namely electrical conductivity, oxidation reduction potential, ORP, pH, and ultraviolet VIS spectroscopy. It is all written in our published article. So this is the way we did it, so that you can see how the information, molecular information, was transferred from the donor to the receiver solution, which was specifically composed. This is the first schedule of our experiments, so that you can see these were different experiments of exposure duration or time, so that the measurements could be done immediately after shaking, or 1 hour after the separation, after this shaking. This is this situation. Or the receiver liquid was exposed to this donor liquid 1 hour after shaking and then separated, and so on. So these were different possibilities. And the one possibility is about the overnight experiment, which was done without activation. This means without shaking, to see what the field itself can do, and if there is any difference between shaking and not shaking. If we come now to the results themselves, we can see that the exposure to receiver, to the donor, did not show any true difference if the distance is concerned. So if they were together, like shown in the previous image, like this is 0 centimeters, there was some effect, it's in average Cohen of physicochemical measurements. Then if it was in the air 1 centimeter of distance, you see this, the effect was almost the same. And it was a little even slightly higher effect if they were separated for 4 centimeters. But all these effects were lower than the effect of the original solution. So this is the measuring of the distant effect. Then if we see the time effect or duration of exposure, we can see that in physicochemical measurements, this is again the average Cohen, average effective size. These are light blue columns. You can see that through higher duration, this effect is raised, while with spectroscopy, it is not the same effect. It drops down through time. Then there is an interesting phenomenon that after 1 hour of separation, the effect if in conductivity dropped to 0, while it was quite big just after the shaking. And then I think this is the most important, perhaps, of all this. So we saw a negligible effect if there was no activation, no shaking, let's say 1 hour of exposure of even 1 additional hour after separation. And there was quite a big conspicuous effect if this solution, detection solution, was exposed overnight. So we see that somewhat this molecular information was transferred to our detection liquid through the night, but it takes time. So it seems that this near field effect needs time. Yeah, so I can come to the conclusions. Now, I think that this finding, this last finding, suggests that shorter exposure times do not necessarily result in a spontaneous molecular signal transfer from one fluid to another. And this is important for the control sample placement. Mostly the researchers avoid the control samples placed near the samples of homeopathic solutions. However, this could lead to exposure to different temperatures or electromagnetic environmental conditions. In conclusion, so we can say that it is still possible that this information somewhat resides in material components. However, through this transfer effect, it seems highly plausible that the molecular information of homeopathic solutions resides in specific energy formations like coherent domains or a sort of easy water or some clusters and so on. The evident possibility of transferring this information via glass and even air tells us that energy apparently locked in coherent domains also has a non-local aspect, which is conformity with quantum mechanics. Thank you for your attention.
Okay, Professor Igor German, for presenting those researches that makes our understanding or giving some clues on how water works on this level. Very important. And now to continue, because it's becoming late to some of us, but early to others. I would like to invite Harry Jebs from the Institute for Frontier Science in California. Harry is going to present to us the research on the unusual properties of water such as easy layer formation, molecular water clusters, and its energetics content. So, Harry, you have approximately 8 minutes for your considerations.
Okay. I'm sharing my screen. Slideshow. Okay, can you see my first slide?
Yes.
Yeah.
Okay, great. Well, thank you very much for inviting us. I'm presenting Dr. Beverly Rubik's and my own research that we did in our um, Independent Institute for Frontier Science in Oakland, California. And what I would like to talk about today is um, our research that we did working with the easy layer that Dr. Pollack presented in his very nice presentation at the very beginning. And I have to say, once somebody does the experiments themselves and looks at them, it's amazing to see these things in action. It's one thing to see them in the presentation, but it's another thing when one actually does the experiment and looks at it. It's just mind blowing in my opinion. So all of this goes back to the water molecule, of course, which is polar as we know, and it has so it can form hydrogen bonds between the water molecules. So water is not just an amorphic um, collection of H2O molecules floating around randomly. Um, if water is left standing for any amount of time, then the hydrogen bonds will form some structure in the water. And here's an image that shows how different clusters can form. So it's not just one particular cluster that can form, it's a variety of clusters that can form. And we believe that each cluster or the composition of the various clusters in water may exhibit various macroscopic physical properties of water. Here's another uh, graph that shows how the nanostructure of water may look like. This is, of course, just a model because nobody can really see atoms, only the response of atoms to certain probings.
Harry,
Harry, could you
just a
please?
comment.
Yeah.
Yeah, your presentation is in the first slide. We are not seeing you. You should put in presentation mode, please.
Oh, I thought I did. Slideshow.
Now we are looking to the flotation water bridge, floating water bridge.
Okay. Slideshow start from first slide. Okay, so now I'm on the first slide in presentation mode, and I'm now forwarding. Can you see it? So can you now see the floating water bridge?
We are looking to the floating water bridge, but yeah, but it seems that you are not in presentation mode yet because we are looking
Oh, I am.
to the other slides. But okay, we can see the floating water bridge. You may go ahead.
Okay, this is really bizarre because, okay, right now I'm out of presentation mode and uh, sure. And now I'm in presentation mode. Can you see now the floating water bridge in presentation mode?
We can see the floating water bridge. I'm not quite sure if it's in the presentation mode, but I think you should go ahead. And
Okay, then something is not quite working. Okay. Excuse me. So then I'm here. This is an actual image of where we produced our own water bridge. It may not be quite as straight as Dr. Pollack showed, but still, you can see when you pull these beakers apart, you can see that there is obviously the water is defying gravity, or so it seems. It's really phenomenal when you actually watch this. Here we have a Nafion wedge in the upper right corner. And under the microscope, we have a water solution with microspheres. And you can see the white exclusion layer that forms. And it's, again, fascinating to watch it under the microscope as it develops. I don't have an animation, but Dr. Pollock showed a nice animation of it. And here, this is a picture where we used, instead of water with silica microspheres, we actually used milk. Milk, of course, is an emulsion of water and fat globules. And here you can, we found that there are uh, fascinating striations of various things that we have not further explored, but it shows the complexity of the easy layer, especially in this case where we use a natural um, colloid like milk. So we thought that was
Harry,
fascinating.
Just
Yes.
another comment, because we continue, at least for me here in my screen, it's with the floating water bridge. I don't know
Oh,
why, but we cannot see the next slides. It's stopped. Now we can see the easy zone. Yeah, the water near membrane self-organizers. So, but
okay. Then
for some reason
somehow
it's
when
not
I
going down. Yeah, go ahead.
okay. So can you now see the raw milk near membrane wedge?
Yeah, now I can see.
Oh, okay. So then the problem is if I am in presentation mode, then it does not forward. So maybe I can do the following. I have to go back. So can you now see the water bridge?
Yeah,
Okay.
yes.
If I now go into presentation mode, can you now see the water bridge in presentation mode?
Yeah, we can see.
Okay, so I have to
No,
go in
no,
and out of present.
we don't see it in presentation mode. So I guess you should either use this or decide to share a different screen on your computer. But maybe you can continue just not screen sharing and scrolling through your slides.
Yes, I
Yeah,
think that's best because we're running out of time. Okay, so here's the Nafion wedge that shows the easy layer that we produced under the microscope. And here is what we produced with milk. We thought this was really fascinating because it shows the striations and the complexity of the easy layer. Um, Here, of course, this is how Dr. Pollock showed it, that we have the easy water and it is charged negatively and the bulk water is charged positively. And that is certainly one of the physical reasons why we see many of the unique properties of the easy water. And now I would like to show some experiments that we did with high voltage electrophotography, which is done by the Russian gas discharge visualization camera, which is essentially an electronic version of the Kirlian photography. The way this works is, excuse me, the way this works is we have a hanging drop of water above an electrostatic plate, which is briefly energized with high voltage, something like 10,000 volts. And then it momentarily produces glow discharge, which is then um, detected by a camera, and then through some minor data processing and images being created. So here, this is the camera. This is an older model, but this was actually one of the first models that came out of Russia. And this is a representation of municipal tap water. So I want you to focus on the center of the image, which shows a certain amount of glow discharge. If you use water from a mountain stream, you can see how much larger it is meaning that the potential or the energetic content of the mountain stream water is so much bigger. And you can also see that there are streamers, which are much more numerous and much more intense in mountain stream water. You can also use an alkalinizer, which runs water through a positive and negative plate, and you take the cathodic water, which is electrically negatively charged, and if you put it under the GDV camera, then you see that it also produces an image with very vibrant streamers, much like mountain stream water. And here we did water that was treated with meditation from a group that was 1,000 kilometers away from our laboratory. And the group focused on one beaker that we had, and we compared it to a control beaker, and you can see the difference. So this is something that is not easily explained by conventional method. In fact, I don't know any method how to explain this. So this is clearly showing that distance is not necessarily an indication for psychic energy transfer, and it can be transferred to water. And here we show how how energetic water can be energized when a Reiki healer it does a spiritual healing on water. And here you see the difference of unblessed water on the left and blessed water on the right, where it's much more coherent and is less jagged in the image. And here we have tap water on the left and tap water imbued with love on the right, which makes a huge difference. So love obviously is a very strong emotion, and it has a large energy content, the nature of which we still need to research. And that it is. I'm sorry that my presentation did not work so properly in the beginning, but I hope you still got an idea, especially from the gas discharge visualization camera, how water can be energized through psychic or emotional energy. Thank you very much.
Thank you very
And
much,
now,
Harry. Yes, we could have a good image of the research you're conducting with Beverly Rubik in California. We are going to the final part of our presentation. We just have one more lecture, and I I think would be important that I share some advances and some actions that we did last year. So just to inform everybody that is listening to us and looking to those nice presentations, that we last year, last March, March 2023, we had the water conference in UN headquarters in New York. And by that moment, some of the colleagues that are here in this event, they signed a cooperation agreement. We're working on advance on this cooperation agreement, and we launched the New York Statement, talking about the importance of the researches related to water on this molecular level, yeah? And little by little, we are advancing on those researches and strengthening this network and this ecosystem of research on this field. And we are planning just after water conference that is led by Professor Pollock, that is going to happen in October in Lisbon. The idea is that we make a visit to Paris, to the PHI, the International IHP, in the International Hydrological Program for UNESCO, to advance for a partnership with UNESCO on this theme of the researchers on the molecular level of water. So just an update, because we're talking about science, but we are also articulating ourselves in this institutional level that is important to have to give those steps in order that those researchers have more strength, more funding, and more priority in all the countries. So just to make this remark, and I would like to invite our last speaker. Then we are going to have a moment of questions and answers. If we have some questions and answers, that is going to be led by Professor Leonie Bonamin. But now I'd like to invite Michael Bash, that is a senior scientist at the Danish Technical University and a technical engineer from the technical engineering department. So Michael, you have 8 minutes. Welcome.
Thank you. Thank you, Sergio. Thank you very much for your enthusiasm and the invitation. Let's see if it managed to go into presentation. It should be okay now.
Yes, we can see.
Okay, good. Okay, so basically, I have a long history of trying to understand water, and I'm just going to give you like 8 minutes of what I currently have achieved so far, and it's always a progress. So nothing of what I say is the final truth. This is just my take on it. So my take is basically that what we call hydrogen bonds are not they're not classical bonds. We're probably more talking about an electrostatic discharge, like we see in this picture of lightning clouds striking over water, like a classical example, and a very good analogy of what is actually going on molecular-wise. So water is oxygen and hydrogen, both gases and liquids at extremely low temperatures. So when you mix them, then you have something that is very stable and takes a lot of energy to evaporate and boil. Actually, if you take hydrogen sulfide, which is very closely related to water, H2S, it boils at -60 or -63, I think. So it's regarded as water should actually be boiling at -60. It does not, because of what we call the hydrogen bond. So that's the glue. This glue, however, is very, very short, and it has two speeds. And these speeds are very, very, very short. It's from the 0.7 to 3 picoseconds, and another a bit longer speed between 5 to 18 picoseconds. Maybe this is a model of coherent, non-coherent water, or high density, low density water. We just don't know. We know, however, that the bond is 11.3 kilojoules per mole. This is too weak for any covalent ionic bond. It is also too strong, what we call van der Waals diffusing bonds. So this is a quite special bond, and therefore I think also the proton in the hydrogen is actually not localized. So you could say that the one electron from the proton is giving off to the sphere, electronic sphere of oxygen. And when the proton is also not localized, this is starting to look like something that is not a classical chemical bond. So so my department is doing a lot of modeling, and I think personally, I think that using a chemical bond approach might be not so correct one. We can see this effect in very weak very simple electrostatic games. Like when you take something electrostatic, you can actually make the whole water column from a faucet bend, and much more than mechanically blow on it. So this is a weak ones. We have seen also the high power electromagnetic effects of water. You put a 10,000 volts, and then you can make a bridge on it. This is comparable to what you have in your own cells. So this is not a joke. This is not a gimmick. This is actually a very good model for what's going on in our own cells. So biology is based on this electrostatic nature, and water is a central role in this. I have personally played around with some devices here in Denmark that I use to remove the chalk from water. We have very hard scale water because we're basically situated on an old seabed. So we have problems with this. And if we apply a very small pulsed field it has a sound component, as you see here. This is the sound component, the wavy sound, and it also has a periodic electromagnetic component. Anyway, what happens is that it goes from this calcite formation, which is the hard scaling of chalk, into aragonite and a little bit of basalite, and that's more powdery. However, we don't know how it works. So I exposed some drops of water from this sound device. And then what you see here on the left, you see is the drying out of a water droplet on a metallic nanostructure surface. And you see a very fractalized evaporation pattern. However, when you have exposed it to these repeated periodic signals, then the drying out of mostly chalk here is actually done in more controlled, more periodic state. That means that the charge of what you had in the water is probably more unified. I also took some scanning electron microscope of it, and maybe it's hard to see online, but on the left you have untreated water, you have many little seeding starts, and on the right you have the treated water. It looks like a branch from a tree. It starts somewhere out of the picture, and then you have a flash deposition of your residues. And this is also an indication that something has been done with the particles that was suspended in the water. On your right side here, we have the zeta potential, so the charge. There is an indication that it's a little bit higher charge, but it's still in the not black and white. We're not really sure about it, but it is an indication of the charge. Now, the big question here is the device affecting the water directly, or is it affecting whatever is in the water? I mean, does it add charge to what's in the water? I think both things are in game now. I have also tried, like Rumiana Tchenkova, I'm also very fond of near-infrared spectroscopy. And this is an old method that has been used since the 60s. When I did repeated measurements on the treated water, this is the green graphs, then they all bunched up below or off from the non-treated ones. You have a little bit of different types of water. There is some tap water, there is thin red line. There is some ultrasound water and some degassed water. And what was common is that it seems to be so that if you treat the water, then you really have something fundamentally different here. So actually, it's the water, baby. It's changing, and this is very interesting. I think it's very interesting from the whole industrial perspective. You could say that the current worry now is, okay, does the Wi-Fi expose us to electromagnetic noise? Does that expose us? But why not turn it the other way around? Could we play with this? Could we actually make a better filter, better pumps, and so on? And not only this, should we also maybe try to look at our industrial installation and realize that maybe having a very strong electromagnetic motor running right under the filter might actually affect our system negatively or positively. Yeah, then here at the end, I have two small examples. This is not my work, but this is just an example of you can use these fields to push ions in the water into the sides, so you're going to have a little bit cleaner water and so on. So this is just an example of electromagnetic effects being used currently to clean out water. We are still a bit far off compared to reverse osmosis, but I think in unison we can get there. Another nice one is to use graphene, which has nullification of the dipole moment, the polarity of water molecules. It's getting broken down near graphene. And this is also an interesting method. In this case, they use it to accelerate evaporation of water in a simple beaker. So quite simple stuff, but based on a very complex nature of water and something we have not fully understood yet. So thank you from here. I have two little recent articles I wrote, and you're welcome to write to me or to my collaborator, Henrik Bohr. Thank you.
Okay, Professor Michael Bash, thank you so much for your presentation and also giving some indications on the practical use of that. We are doing this event in parallel to the World Water Forum in Bali, and it's important that water sectors, engineers, and people from the water world understand better the possibilities and the potential of understanding water in this molecular and nanostructural level, yeah, in order that we can advance on this area. And now to close our event, I would like to invite Professor Leonie Bonamin. She's a full professor of the University Paulista, São Paulo, Brazil, and she's also a member of the GIRI, an International Scientific Society for High Dilution Research. So, Professor Leonie, you have the floor, and then you can close the event this night or in the beginning of this day for some colleagues. Good night.
Okay, so thank you, Sergio. I can't see any question in the chat, so maybe you can move to the closing session because you are late. It's all right, Sergio? Can I close the session?
Yeah, please close the session because I asked to the team that are supporting us. There are no questions until now, so you can go ahead.
Okay. Just to talk some words about what we could see today in the session. First, we started with the conference of Professor Gerald Pollack. That's always a pleasure to hear him talking about all his experience with water properties and new discoveries about water. And it's interesting to see how this subject is indeed in the frontiers of science. So we have new things, old properties of water, but new discoveries. So we are just learning about these properties, but these properties were already there in the world. So this is amazing to see, and how this subject is evolving in function of time, mainly recently. And after we could see fast talk from our colleague Evelyn van der Kras, and she presented some words about the World Water Community and World Water Lab. And she's the coordinator of this initiative, and it's very interesting and important initiative to join scientists and the society and the industry. So with this resource, I think that basic research we are doing about water and high dilutions and all our other related aspects can be fastly in the society and in improving possibilities to create new technologies in a very fast manner. And it's very important nowadays, facing all the troubles we have in our planet. So it's a very, very good initiative. And after Professor Rumiana Tsinkova, she did a very fast and very comprehensive explanation about aquaphotomics, and it's her field of study, and it's very interesting too to see how this basic research can be fastly applied in different fields of knowledge, including biology, and how it can change our perspective about biology phenomena such as biochemistry or physiology, and it can improve also our understanding of health, and also to create new technologies to deal with health and health issues. And then Pierre exposed about some flashes regarding the special edition of the International Journal of Molecular Science that he was in charge to organize everything. It was a great also opportunity to bring the idea of coherence domain and how it can fit different aspects regarding biology, and mainly a new perspective about hormesis, not only under a strict pharmacological aspect, but also biophysical aspects. So he could explain several examples here, experimental examples, showing this possibility. So it's another new subject in this field. And then Professor Igor Yerman exposed to us different experimental models used to understand the physical properties of high dilutions and the scientific basis about the effects of high dilutions, and including homeopathy and some related aspects. And then Professor Harry Jebs showed us different models too to study water physically, and with a new element that is the GDV technique that is seems to be a promising technique, but it still needs to be reproduced by other researchers, and this is a technique that must be improved. But the results presented are amazing and very interesting, and it must be taken seriously because it's a very, very important and serious subject. And finally, Dr. Michael Craig showed us some technological solutions using all this knowledge and put it all together in more or less simple elements, but that can be used in a very, very short time. For example, desalinization and other applications that are very, very needed nowadays. So it's beautiful to see how basic research, studying water in laboratory conditions, passing through biology, health, and other elements, and then we can get a very, very applied possibility to use all this knowledge using sustainable, new, and interesting and affordable technology. So we are in the edge of the future with this theme. So I'd like to thank everyone to be here and our contributions. And so I'd like to close the session. And Sergio, if you want to say something, please feel free.
No, just to thank once more all the lecturers. I think we had a great night. We are trying to strengthen the group. This is the third edition of the World Water Forum that we are together. We were last year, as I mentioned before, in New York in the Water Conference, UN Water Conference. So what we are trying to do is to, as I said before, to strengthen this network of researchers, to gain more visibility, to gain more support, in order that we can advance on those researches in a faster way and with more depth. So I'd like to thank everybody that was with us during the about 1.5 hours of event. So thank you so much. We are closing, and we see each other soon in another webinar. Thank you.
Thank you, Sergio.
Thank you.
Goodbye.
Bye-bye.
Bye-bye. Thank you.